Welcome! Today we are going to wire up a simulated retina, inspired by the human eye, to a small visual cortex and a handful of other brain regions. We'll give it a body, give it the ability to learn, drop it into a world and see what it does. So, before we get to the neurology, which will be covered in great detail later in the video, let's look at the world these creatures will live in. The environment is built in Godot and Blender, and I was careful not to overload it. It's an experiment, and I want it simple enough that multiple creatures can render their own view of it in parallel, and that's why performance became necessary to pay attention to. As you can tell, the assets are all handmade and low poly to keep rendering fast, with specific visual cues that creatures can learn to recognize. Everything is placed deliberately. Each creature sees the world from its own perspective. Godot caps me at 64 active cameras, which left me with the choice: 32 creatures with 2 eyes each, or 64 single-eyed pirates. I went with the pirates. Even though I initially built the character model with 2 eyes, that's fine, because they have no idea what eyes even are. It would be cruel to pop them into existence with nothing but a static map, so I built in a few ways for them to interact with each other, enemies and objects in their world. Let's look at the map. The map is split into 3 regions: a desert, basically sensory deprivation, a forest where the creatures can thrive, eat and play with each other, and then the bad guy half of the map, which you can spot by the badly drawn volcanoes and big magma lakes. All 3 regions are connected by passages. Designing the world required strategic planning. The brains controlling the creatures are small and will almost certainly not be able to learn complex features, but they still need visual cues to tell objects apart, so coloring and shaping the world had to be approached by putting yourself in the network's shoes. The world has a day and night cycle, making sure the visual stimuli changes even if the creature is standing still. There are enemies, the red creatures, that randomly kick the blue creatures around and also eat their food. There are simple programs with no brains, so they are predictable. Don't worry about the blue fellas, they don't feel any pain. It might even be kind of fun for them to get thrown around and stuff like that, because whenever they are off the ground, a specific region of their brain receives excitatory pulses that trigger what I call their instincts, taking over and producing movement actions. That's not happening right now since I'm controlling them for demonstration purposes, but don't worry, at the end of the video you will see the first bio creatures and watch their world from their eyes. They can also swim, which triggers the same excitatory pulses as falling or flying. Their perspective is, of course, from in front of the body, which protects their innocent soul from realizing how goofy they look while swimming. As you may have guessed, I tried to make a few activities fun for them, but they had to stay simple. And because the red guys are kind of bullies, I created safe zone bulbs that the blue creatures can punch. These light up the area for the next 5 in-game minutes and scare the bullies away. Doing this isn't tied to any reward, so I was curious whether they'd ever figure it out on their own. Their food is a yellow pinkish mango. It has unique colors in the game and a pulsing glow, and it occasionally twitches to catch the agent's attention, like a firefly in front of a frog. So there's a working food system in the environment. The creatures can also punch enemies, just like enemies can punch them, and while the blue creatures are immortal, at least until the end of the simulation, the red ones aren't. A few punches is enough to make them disappear, but they will respawn after a few minutes. Here's the most challenging task they can accomplish, and the one that triggers massive dopamine spikes telling them that what they just did was right. The altar. They can sacrifice the ever-spawning, ever-despawning balls in it. The altar tracks every contributor that brings a ball or balls into the altar, so each of them gets rewarded when the task is completed. They can also fail and the ball can roll away, so this is a task that requires either a lot of luck or deliberate action. Once it's done, a secondary reward spawns. 4 mangoes inside a ball. The sacrificed balls disappear and respawn elsewhere. The altar has a unique shape in their world. I choose the same color for the ring as for the creatures, not to trigger any religious immersions, which would be hilarious to be honest, but we are not on that level of complexity yet. I just wanted it to have this distinct ring shape the brain can recognize, with strong contrast against the environment. The game itself is only part of the system. I needed a remote control and it broke nearly everything at the start. But luckily, through the magic of video editing and a ritualistic sacrifice of a huge chunk of my time and sanity, I was eventually able to control all of them individually from a single Python script, with just one instance of the game running. It was a huge challenge because pausing the game broke the physics and I had to add a lot of checks and clamps to keep the objects from getting stuck in space. I won't bore you with the details, but I'll give you an overview of the environment. Let's talk about the brain, the star of this whole project. And we'll start with the eye. Right now you're looking at the model in 3D, and this thing has a few tens of thousands of neurons in it. So, how is it built? The retina stacks into 5 classical layers. Photoreceptors at the top, then horizontal cells, then bipolars, then endocrines, and at the very bottom, the ganglion cells, the ones that send the signal out. Almost every layer is split into a foveal part and a peripheral part, and most of them are also split into on and off channels. Here's how an image gets in. It's first split into 3 channels, one for L, M and S cones. Those channels get sampled at the positions where the cones sit, but the cones themselves don't actually spike, they are just position markers. The sampled values run through a biophysical phototransduction cascade, basically an ODE system that approximates what real cones do with their cGMP and calcium chemistry, and the output gets compared against the spatial mean across each bipolar population. The difference is what's injected as current into the bipolars. That's where the real spiking starts. So, right now you're watching the neurons of the retina light up as a video plays on it, in slow motion. The first spikes come from the bipolar cells, which get the photoreceptor-derived current. And then horizontal and endocrine cells join in. And finally the ganglion cells at the bottom. From here, the retina projects onward, with the midget, parietal and conio streams kept separate, roughly matching how the primate retina hands off to the thalamus. I went through many iterations and a lot of calibration work and eventually settled on a configuration where most populations fire within the rate ranges reported in the literature. What you are seeing now are spike rasters from 3 of those versions. The visual pathway is almost entirely built in neurotics. The brain is basically 2 things. A retina, the red part, and a massive graph that keeps track of every population, every connection, and every wiring rule before the network even spins up. From the retina, the signal lands in the LGN, a relay station in the thalamus where pretty much everything heading for cortex passes through. That thin layer next to it is the TRN, a kind of gatekeeper, and every LGN channel sends a sister projection through it as it goes up. Then comes V1, and the layered structure actually matters here. Motion lands in one sublayer, fine detail in another, and color in the third, all sorted from the very first cortical stop. After V2 we get to V3, which is the fun one, because every cell type gets a dorsal and a ventral copy. The ventral side commits to V4, while the dorsal side goes to V4 as well, but keeps going into the pulvinar, into V6 and further out. It's not a clean dichotomy, more like a scaffold for one. From here, the brain picks 2 sides. V4 is the what, it talks to memory and value. V5 and V6 are where and where it's going. V5 wires into motion primitives, V6 into head direction, and sitting between the streams is the pulvinar, a thalamic hub that lets them whisper to each other. The rest just closes the loop. Superior colliculus and the frontal eye fields decide where to look. Basal ganglia wait in what to do. And the hippocampus try to remember any of it. So, this took me a few months. Reading papers, watching lectures, trying to turn all of that into something that actually runs. I can't honestly claim I reproduced any specific finding, but every population and every connection in here is placed by hand. Is this a primate brain? Definitely not. Is this a defensible scaffold built from what the literature currently tells us? I can't tell. But it does things. Some populations' activity depend on the size of the network, some populations fire very rarely, tweaking parameters on one side of the network can lead to bursts in another part, and without the intention of this being an excuse, this is version 230. I'm a solo developer and it works well enough that I can start showing first results, even if the network is not 100% operational. One quick thing. To preserve the spatial layout of the visual signal, I'm using flat sheets of neurons as terminals. Small grids of 32 by 32 cells. This was inspired by the way the retinal signal projects retinotopically onto the visual cortex. Building sheets of neurons was the next best idea I had to approximate that, because they allow for structured one-to-one connections. They receive the input in the right order from upstream and then fan it out into the rest of the population. They are not the only kind of connection in here, but I wanted you to know the visual signal comes through structured. If you are wondering how we are going to have multiple creatures, here's how. They all live in the same simulation, separated by an offset, completely disconnected from each other. Each one has its own retina, gets its own inputs, and is fully independent of the others, but tied to the same timeline. The whole setup looks almost like classical reinforcement learning, with one twist. The environment generates the sensory input, pauses, and ships it over to the networks as a batch, where it gets distributed across each agent. The networks then keep simulating, just with the new input baked in. After a fixed amount of in-simulation time, the networks pause again, and we count the spikes at the regions that are mapped to actions. The ones with the highest spike counts get picked, batched back to the environment, which executes them, updates the world, generates the next frames, and the cycle begins again. Let's look at the building block of action selection. It's made of 3 parts. On the outside there is a liquid network, a recurrent pool of neurons that mixes the signal in time. Sitting inside is a cone, a bidirectionally connected cluster that projects into the actual control block. That's where we count the spikes. We built a 3x5 array wired so contradictory actions can't fire at once. We then assigned actions to the control populations. 2 pathways feed them. The liquid gets weak learnable inputs from V1, V2, V4, V6 and S3 atom. The other goes through premotor blobs. V5 drives movement actions, V6 drives head direction, V4 and V6 together drive hand directions. The controls then mix this with the frontal eye fields, the primate's eye movement system, here also steering the head. And if a reward is collected, some of these connections will get dopamine spikes and the network learns. I use 5 main types of synapses here. Time to see the neurons fire. The retina and the network are freshly built. Right now it's looking at a slow motion video of an orange cat, purely for demonstration. To the network it's just a bunch of random signals. But I gotta admit, it feels awkwardly funny to show something freshly built a cat video. Feels right somehow. Okay, as you can see, the first spikes are arriving now. The real retina does a lot of pre-processing before sending anything onward, and a simplified version of that happens here. This is the LGN firing. Populations that belong together are stacked together. Some of them need a warm-up phase, and since this is slow motion, a few might look completely silent at first. I learned not to trust the first 20 seconds to 3 minutes of the simulation because some dynamics depend on already functioning substructures. Above the LGN sits the TRN, and then comes the first big cluster of neurons, V1, which reacts directly to the visual input. Every population here has its own parameterization, so they all behave a bit differently. Some neurons are fast spiking, others need a build-up phase before they really start firing. Timing matters too. We are moving past V2 now, and V3 is starting to wake up. Some chunks of the hippocampus and the basal ganglia are on screen as well, and right above that sits my attempt at a custom motor system. It also needs a warm-up. At this point, we'll wait another 10 to 15 seconds for the visual signal to finally reach the motor system. What you are seeing here is the network after 2 minutes of video. Most of the populations are active, but it hasn't learned anything yet. Its parameters are tuned for long-term learning, so even an hour of video won't make much of a difference. This thing learns in days. At the start, it picks essentially random actions, but over time some connections grow stronger, others weaker, and through the STDP synapses, it should slowly learn to process the visual input. I've been wrestling with myself over whether to release this footage. Most videos like this only show the wins, and only a handful of people have the patience to actually listen when someone talks about what went wrong. But you are still watching after 16 minutes of me talking about a niche topic, so I'm going to assume you'd like to hear about that too. If not, feel free to skip ahead. The first footage of the creatures is at this timestamp. For this project I had to build intuition the hard way, by experimenting, and in this section I will share what I picked up along the way. Because honestly, it was the failures that taught me the most. Some of the early approaches were incredibly naive. Parameterization was chaos, and topology was a mess. The first attempts had an arbitrary sized LGN and V1 through V6 connected in some simplistic way, with crazy connections internally and across the areas. You could call it an attempt to get the signal through, but it didn't get through. Feedback loops, bursting populations, dead populations, just a few of the problems that didn't go away for a long time. I tried to model the visual pathway schematically, placing connections like arrows. This goes there, this belongs here. That was the whole logic. I didn't expect any of it to function. I was aware of how simplistic the approach was. The interesting thing was that the system actually got more stable the closer I moved toward higher complexity. Sure, I had plenty of network meltdowns on the way there, but the trend genuinely surprised me. You might say, duh, the closer you get to what biology actually does, the better the model behaves. But it really did surprise me because I'd expected the opposite, that the more populations I added, the more unstable and unpredictable the whole thing would become. Sure, the amount of parameters grows, but in the end there are only so many populations you have to pay attention to. It's doable. I was glad to push past that barrier where you keep fixing small things until they work, only to realize later that the interplay between the parts doesn't. But it had a price. I had to rebuild everything, and I gladly did, multiple times, adding more and more biology to the structure. But this trend might be only true for my case. What I also learned is that the size of the network matters. There is a certain scale that doesn't apply universally, but specifically to the task you are trying to solve. Put too few neurons into a population and you won't get the right population dynamics. Make it too big and the same parameters turn your network into a lightning bolt or a dead cluster. Inhibitory connections often work well when you weight them about 3 to 5 times higher than the excitatory ones coming into the same target. Depending on the task, of course, but that's a magic number I remember. Slow down the learning process and clip the maximum weight any connection can reach to a value that feels right. Ask yourself, once the network is fully trained, how much power should the signal carry when it passes through this part of the brain? Here's an important one. Populations that on their own seem to behave one way, sometimes even boringly, can turn into absolutely interesting functional building blocks once they sit inside a bigger structure. But don't let small-scale configurations fool you. Wiring seemingly uninteresting topologies together often results in very complex behavior of the merged network. Sometimes it's useful to think of populations as gates. But that intuition breaks down both below and above a certain size. It depends on the neuron parameterization and connections, of course, but for me the sweet spot for one block sits around 1000 and 4000 neurons. And if your network looks stable, test it for longer. Sometimes stability comes with time. Sometimes a network switches between chaos and order. Sometimes populations start to burst seemingly out of nowhere and destroy other dynamics around them. One thing I heard a lot from the community is to build your system subcritically, or even sub-subcritically. But here I found I had to stay closer to the biology. The real dynamics only seemed to emerge once populations were woven into the bigger picture, and that meant living near the edge of chaos, not below it. This is also what I meant earlier when I said populations on their own often don't show the fine dynamics they develop once they are part of a larger structure. But before we end this section, I want to share something I found beautiful. And I'm about to get a little philosophical here, so take it with a big grain of salt. When designing the liquid network, I stumbled upon a few configurations that showed these interesting group dynamics. Dancing signal patterns moving through the population. It was mesmerizing to watch. And I couldn't help but theorize that what I was looking at, these dancing patterns, might be something of our kind. Not a consciousness, but an object, an activation pattern related to the thing our mind is, like a stone is to a mountain, that everything you perceive, if you drew it schematically like this, would have its own dancing pattern, a signal shaped by its own environment, trapped on the biological highways our brain uses to control it, a fire spread exactly where it's needed and contained so it doesn't burn everything down. It was a beautiful and tempting perspective, but the brain is so much more than that. One last thing before we get to the first results. This project became more ethically challenging than I initially intended it to be. The scale at which something becomes able to suffer is not clearly defined, and where sentience begins is still a highly debated question. But at this scale, a few tens of thousands of neurons, roughly a tenth of a honeybee's brain, I would argue we are far below the threshold. That will, of course, change as we scale up and start building more complex circuitry, and at that point, these questions will have to be asked seriously. For now, my claim is that at this scale, these creatures will not experience agony, as long as their existence itself doesn't cause any. They are, of course, trapped in their world with no way out. But then again, aren't we all? Anyway, computing a few seconds of their life took hours on my PC. The software is obviously meant for clusters with a load of RAM. This is what their first few seconds of life looked like. Every action is chosen by the network, randomly at first, of course, and that won't change in a matter of seconds. They are basically newborns and have no idea whatsoever what's even happening. Yeah, here I tried to reduce the computing time by reducing the resolution of the retina and shrinking the network. The network stopped working at that scale, so we have a minimum size with that parameterization. One slight turn to the right is all we get. I was first confused why this happened. But then I saw this. Alright, so that's it. I've run out of video material and I think this is a good place to end. Thank you so much for sticking around. Have a nice day and I hope to see you in the next one. Bye.